Good evening, everybody. Welcome to Taxonomy Tuesday, where we help each other find out what we found while we were out there and discuss the likelihood of what it may or may not be and correct anything that needs correcting and see what we have. If anybody has anything they'd like to share from their machine, just put it in a chat and we will get to it, or just shout out if you have anything. I know we had a little mail from Shihang, but she's not here yet. And I haven't seen anybody else mail in.
I have stuff.
You want to share from your machine?
Yeah, I can do that eventually, or now, or yes, whatever, whenever.
Go for it.
One moment.
There you go. We're seeing stuff.
CI now?
Yep.
Great. Okay. So, I found my first Lactifluus for the year. Lactifluus volemus. So this is a Lactifluus, which used to be in Lactarius, and meaning it lactates a lot. So the ones that give out a lot of a milky substance went to Lactifluus, and this is Lactifluus volemus, and it has a white milk that stains brown, and people say it has a fishy smell, although I think it has a really good kind of sweet smell consistently for me locally here. So but I'm pretty sure this is what it is. I did do some microscopy, and these are the spores. They're pretty neat, very heavily ornamented, and they kind of look like cannonballs or
That's a nice shot. Is that just in water?
no, it's in I think it's actually a little Congo red and water, I think.
Can see some good detail on there.
Yeah, so I mean and it's magnified with my phone quite a bit too. So
Right.
yeah, and when I share like this, I can really blow up my whole screen. I just like take my fingers and stretch my whole iPad screen. So I think that's what helps the most, but I'm not positive. But yeah, so yeah, that was exciting, and I'm going to eat them, but I didn't yet. I actually tonight tried I've tried it before, but I had three of the green russulas. I've never really had more than one that was decent to eat, so I ate kind of a whole little bowl of the caps sautéed tonight. So if I start to act funny or something, I made the wrong ID, but no, I'm certain of their ID. But they were pretty good. They were a little bit towards grainy. The texture was a little bit different, but the smell of cooking them was really nice. It was really reminded me of porcini, but the flavor was not that good. It was good though, but not not as good as the smell when I was cooking them. So yeah, so it was nice to find these, and those were south of me a couple hours, in central Illinois. So and then I think probably everyone's seen these before. Galiella rufa. I love these. I call them peanut butter cups. Um, and I did microscopy on these, and they're pretty cool spores with droplets, and mainly, well, a lot of them had two, but I can see that some of them seem to have more.
Do
So,
these have a little bit of that, like almost like knob end they look like?
oh yeah, kind of almost like the those neogyrometras or gyrometras. Some of those had that. Yeah, it looks that way, doesn't it?
Yeah, a little
Or
bit.
or they're at least like flat ended a little bit, but more like a knob, like you said. Yeah, kind of cool. Yeah, and I didn't check on these, nor the Lactifluus with any resources to, see that they're supposed to look like this. Um, but I imagine the IDs are both correct on these. Um, and then got a couple more. Let's see. Um, this Cortinarius, I've decided is Iodes. Apparently, it's it grows in oaks, and this was an oak woodland. So there's Cortinarius violaceus and Cortinarius iodes, and then a couple other purplish quartz that look pretty similar. But I think that this is iodes.
No, it's not.
It's not.
No.
Okay. Um, and we can talk about that. I'd like to hear why. Here's the spores. They looked pretty similar to me, to iodes. My first guess was violacea, but then I think that's more conifers. And then I was able to see
I
some.
think maybe what Igor is getting at is there's more than these options for just these options for these purple quartz.
Oh, okay. Well, I mean, yeah, there's probably. Oh, well, there's all the ones without names too, like NM102 or yes, correct. Sorry. Yeah, with real names, I think there's maybe four or five, but then with provisional names, there's probably another 10 more or more. Or, I don't know. Um, but yeah, okay. So, well, I love to hear people weigh in. Um, but this, I assume, are Pisidia with spores on stigmata, stigmata.
Yeah, I'm digging that. You could see the all four on that one.
Yes, that's neat.
Yeah, it's pretty cool. It was fun to work on. But so okay, but I've come to apparently the wrong conclusion. So, Igor, what do you think it is?
No, I know the species name, but this is not Iodes. It's too robust, has the wrong bulb, and Iodes is slimy. It's wet. This thing is dry. I think it's from different
You're right.
section or subsection or whatever, whatever the taxonomic structure of Cortinarius is, or Cortinaceae. This is in the wrong pond.
Okay.
So, but notably, Iodes is sort of purplish lavender, and it develops these lighter, whitish or yellowish areas on the spots, on the cap. This thing doesn't have that. But more importantly, this thing is dry, and it has a very, very
It's
bulbous
dry.
base, and it's very robust.
Okay. So I just will call it Cortinarius, I guess, for the
Yeah,
moment.
well,
Or even.
Hey, could you go back to that shot where they're laying down?
Yeah.
Just for anybody new, the ring around there, that's spores left on the remnants of the tortina. Bud, you can't be doing that. And so that's the spore color too, that rusty brown.
right.
Yes.
The cortina is
Okay.
very wispy in many species, just breaks apart, but part of it is left on the stipe as a ring zone, and that traps the spores that drop down, and hence you get that rusty brown ring where the cortina used to be attached to the stipe, and it just traps like a filter all these spores.
Right. I love that. All right. So back to the drawing board on a name for this. Thank you for letting me share. I think I have one other one. So yeah, we can look at these foliates. I thought they were Hemileccinum subglobripes, um, which which in to the north of me or is found more in conifers, but in my area of Illinois, it's found with oaks, according to Quo. However, this is south of me, still a couple hours, and um, it's been brought to my attention that it's probably not some gobbler peas, and instead it is. Okay, wait, it's not here. Sorry, this would be better. Um,
Yeah.
Let me just get here. So Hemileccinum sutara, and Igor made this ID. It's
Yeah, I
interesting.
wasn't sure this was the Labripes. It's too brightly colored. Has two corrugated cap, and it's just too south of the Mason-Dixon line. Sublabripes is more of a boreal species. It likes to grow with birch in New England. In New Jersey, it's uncommon, but we did find it at Stokes last weekend. But that's pretty much where its extent ends. It's just above the Mason-Dixon line.
Yeah, and I found it in northern Wisconsin in like birch and hemlock.
Yeah,
Yeah.
yeah,
Uh,
you'll find them in the mixed woods. I think it's mostly a hardwood associated species. It might grow with oak, but oak is more rare up north than here. So I just call it Hemileccinum because I'm not sure what species it is. It's not rubropunctum. And, I think I did tell you that there are other players that haven't been properly described yet. There's species Indiana 01, there's species Sublabripes Indiana 01, which are distinct genetically. And I think there are some names from the past that is going to be equated to one of these at least. Basically, they are described species, but obscure, have been forgotten for decades. Nobody knew what they were. There's also this provisional name that I gave to a Hemileccinum, called them enigmaticum, because it is enigmatic in a certain way. So there's quite a few Hemileccinums, and of course there's Hortonii. I don't think this is Hortonii though. Albeit, one of the caps you can see in the far distance, it's quite corrugated, quite wrinkled.
Right. But
So I wasn't sure about the species. I just called it, genus. If you have a specimen, it can be sequenced, obviously.
I would like that. I would like to do that. I do have one. So
But I think they're all edible. I don't think it's been implicated. This genus has been implicated in any poisonings, but it's not a spectacular edible to begin with. I've eaten Hortonii, and I've eaten, I think, maybe rubra punctum back in the day. I don't think I've tried some Labrapes because it's so rare in New Jersey. I've just seen it a handful of times.
I've eaten it. It's very good. I do like it. But I didn't eat this. Yeah. So I will drive this and send it somewhere.
Yeah, sounds good. Because we pretty much know the extent of the genus at this point in time. Everything that people found over the years. Steve Russell sequenced it. Colin Damnauer got the type collections from the past, so I think we're well covered. I think it's a small genus still. Maybe all in all, 6-7 species.
Okay, cool. I think I've stopped sharing, and thank you.
Thanks, Hunter.
My
That's
pleasure.
fun stuff.
Hey,
Yeah, just one quick
Chris,
comment.
did you get a email from me? I sent you a lichen, interesting lichen. It's Doug.
Yeah, I'll grab that, and I think Paul had some. If he was ready to share from his, I'll grab that email.
Yeah, I'm ready. I see Shihong is also here, but I've just got a couple. Maybe I'll just go real quick here. Let's see. Share. Right. Is it? Is it? No. Is that it? That's odd. Is that it? That's not it. Nope. Well, that is. There we go. Okay, I have a bolate, so I'm glad that you're here, Igor. And what's this here? What is all this? Get this out of the way. Leave that there. I used my boletes of North America.
We're not seeing anything yet. I'm sorry to interrupt.
Oh, shoot.
It's just a black screen.
Thank you. What about now?
Oh, yeah.
Yeah. Okay. Now we see some. Okay.
Okay. I used the Boletes of North America book, and I think I've got a good idea here of Boletus vermiculosoides. I found this along the trail side. Oaks and hickories and some various other mixed hardwoods. And when I flipped it over, I was surprised to see this rather reddish orange pore surface here.
Neat.
Brown orange.
Yeah, and I cut it in half, pretty well hollowed out at this point. Visited by some bugs, some bluing, not a whole lot of bluing. And this was about within a minute of cutting it. That fairly quickly sort of faded to sort of a grayish brown. I did some reading up on it, and so I thought it was important to get there's the pore, sort of, I guess that's a black color there. I did do some spore measurements because I thought that might be important. These spores measure around 9 to 10 microns long or so. I understand there's two species. There's a Boletus vermiculosus and a Boletus vermiculosoides. And I think spore size was one of the differentiators there. What else do I have? This is one of my first shots ever of the cap surface, the pileopellis. I think this might also be indicative, although there's some confusion on whether we have what is the term, cutis or a trichoderm type of pileopellis. But anyway, I think that's a correct ID. Any thoughts on this one?
Yeah, this is the correct idea, I think. That name, by the way, is going to go away because it's been found out that another species that Smith described in his book by the name of subgraviolens is the same species, but that was published on a preceding page, or it had basically an earlier page to it. And because of that, vermiculosoides is going to be collapsed into subgraviolens. So subgraviolens is going to emerge as a new name for this species, as Neoboletus, obviously. Neoboletus subgraviolens is going to be a comb nov. Yeah, nobody knows what vermiculosis is until somebody sequences PEX type.
I see. Okay.
So for the time being, we're going to operate with the names that we know.
I see. Okay.
Everything else is a conjecture at this point. So yeah, the bullseye, you got it right. And this boleta is slightly variable, but I've sequenced quite a few of these, and then they all gave the same LSU ITS sequences, and I presume there's just one species. It's just not a species complex. It's the same thing.
See, interesting. Very good.
Yeah, never ate them. They're probably edible, like all the other.
Looks like the bugs think it's delicious.
Yeah, you got to be careful. There's at least one toxic species of Neoboletus in North America. So you have to be, and it doesn't have red pores, by the way. It has yellow pores, surprisingly. So you have to be careful. But eventually somebody's going to try this thing and say it's okay to eat, but it ain't going to be me.
Yeah,
nice find.
My understanding is the vermiculosis name is indicative of being infested with insects. Usually, I think that's what the name roughly kind of gets down to.
Correct.
So thank you so much.
Yeah, you do find these quite wormy and hollowed out, as you said. That's a good word.
Yes. Cool. I've got one other one I found today, which I found before. I think maybe you've all found this one too, the snow fungus, Tremella fuciformis.
So pretty.
Yeah, it is. It's very delicate and so translucent, almost transparent. Well, translucent, I guess is the word.
Yes.
Yeah.
This is near.
We've all seen it before, but I don't see this that often. In fact, I saw it today for, only maybe the second or third time ever.
Oh, is that right? Okay, that's interesting.
Well, at least out here in South Jersey.
Yeah, this one's growing on some sort of. It's probably a tulip branch there, and I'm not sure whether it's a pure saprobe or whether there's also some parasitism going on with this fungus. But I did get one. I haven't I just found it today, and I haven't gotten any spores yet. I probably do, but I did get one shot of these. These are basidia that are, as they mature, they become cruciform or cruciate. They get this sort of plus cross sign in them, and this is a very similar picture to what I see on Mushroom Expert of these basidia. That's the only micro photo I have of that, though.
So are the Pisidia, are these kind of free floating then, or
Yeah,
are they
I,
attached to something?
I did a quick, I cut as thin a section as I could, and it's smashed quite easily under the cover slip.
Hmm.
So I'm not sure what to
Yeah,
tell you there. It's a
yeah,
very,
yeah.
yeah, very squashed specimen there.
I'm
That
I'm
is
Uh,
cool though. You can definitely see the cross or the plus sign. Yeah.
yeah,
I'm
so
seeing a
that's
bunch of them with just a single line. I mean, is the plus sign or cross only in one orientation, I guess?
I'm not sure whether they're maybe on their way to becoming, fully cruciate or what the story is there. They talked about at maturity, having this septation or septate, four septate. So, but that's my only shot. Yeah, it looks kind of like slotted or Phillips head.
I got to tell you that one might be fun for the photo contest because it's just there's a lot. There's a lot doing there.
Yeah. So okay. So anyway, Tremella fuciformis.
And
And
I
that,
think that grows with like, is it hypoxylon or that
right.
black fungus on? There's a relationship with that. For this
That's
to grow, the other one has to be there, I believe.
Yeah, that's my understanding. There's some kind of relationship there, but I don't know much more. I haven't read up. Like I say, I only found this a few hours ago, but I
Yeah,
didn't see any. I don't think I see any hypoxylon on here. I see maybe a little sterium, maybe or something over there, but
yeah.
Some Tremella species, I think Tremella mesenterica is actually a parasite on Sterium mycelium.
I see.
I think. I'd have to look
Okay.
that up. Is
Yeah,
this thing still in
you're
Tremella? Is this Tremella fuciformis?
As far as iNaturalist thinks, yes.
Oh, okay, yeah, they keep up to date pretty well. So it's still in Tremella. Okay, so it's believable this would be a parasite on some wood inhabiting fungus
Mhm.
yeah,
like
most of
Sterium.
most yeah, most of tremolo is. And Hunter, I just looked it up. You're right. It's a hypoxylon for this one. Uh, for Dacromyces, certain species of Dacromyces, uh, that's parasitic on Sterium, and then Tremella mesenterica is actually parasitic on Peniophora, Peniophora mycelium. So a
Okay.
Okay,
lot of these
not
jellies.
Sterium. Oh,
Yep,
okay.
Peniophora.
Hmm.
The Dacromyces species are, and then what is it? Neo, whatever that new species is, new genus is, Nea Tremella, or something like that. That is also parasitic on Sterium, but a lot of these jellies are parasitic on either the mycelium or the fruiting body of another fungus.
I see. Okay. Interesting. Interesting. Well,
Yeah, but
thank
Hunter, you
you.
were right. Hypoxylon archerii for this one, according to mushroom expert.
Hey,
Okay.
in between here, real quick. I don't know if Doug got my chat. I'm not seeing your email, so maybe send it back in.
Okay,
Sorry.
yeah.
Well, thank you very much for the help there, and thanks for hosting, Chris.
Oh, that's fun. You got some fun finds there. That's a good micro. I'm telling you.
Thank you.
Uh, does anybody have anything from their machine, or we could? Shihang is here. We could check out her finds. You ready to go, Shihang?
Yes, I am.
All right, let me pull up.
Okay, so I was hiking in the Dolomites area for 5 days. Mostly it's above tree line. I only saw two mushrooms, so I want to share with you. So this is the first one I saw. Um, I know it's a kind of a agaric, but I have no idea what exactly it is. I put it INAD, and this is the top choice from the AI. Melanoleuca cognata.
I was going to say Melanoleuca. The gills are close and kind of neat looking. It looks like Melanoleuca.
Okay.
Yeah, the straight stalk. I don't know what species to say, but it looks like Melanoleuca. So it would have white spores, white spore print, and if you have melters, the spores will be interesting. Even if you don't have melters, if you have a microscope and you have a stain like Congo red, you can smash them on a little bit of gill. And most Melanoleuca species have really interesting cystidia on the gills. They look like harpoons.
Hmm. Well, I don't have a microscope, and also this was taken on a trip.
Oh, yeah, yeah. So, yeah, even if you had a microscope, you might not have lugged it up onto
That's
this
right.
trail.
Yeah. Good. So at least the genus was confirmed.
That's fun.
I think I sent three. The Okay, so this is the second one I saw. This one I'm pretty sure it's a sweller. Uh, which kind of swellers? I have no idea. And this is above the tree line, actually.
So there were pines up there, I guess.
Yeah, there was a very short pine tree or whatever evergreen nearby.
Right.
Yeah,
Where
this
was the Dolomite? Where are the Dolomites?
I believe this was at the
Yeah. Yeah. What?
Trichemia area.
Oh,
Yeah,
in Italy. Oh,
yeah,
wow.
oh, this is from Italy. Oh yeah, it looks like granulatus to me. I thought you found it here in New Jersey or something.
No, it's not in New Jersey. We don't have anything above the tree line.
Yeah, well, yeah, I concur with that idea, but I don't know European Suillus that much. But I've collected Suillus granulatus, bona fide one, because it was sequenced in
Okay.
Lithuania a number of years ago. So it kind of looks similar to this. So I think it's probably correct.
All right. So it was funny that I uploaded, I put in a lot of wildflowers. It's like I upload during the hike. In the evening, I can already see people comment, but for mushroom, nobody commented at all. So there are much less people into mushroom than people into wildflowers.
I think that
What a
holds.
shame.
I think you'll get some responses on the flowers. It just takes a little while.
Yeah.
But that holds true for here in New Jersey as well.
Yeah.
There's many more people interested in or that know flora and fauna than they do fungi. So anytime I post a bug, I get a bunch of responses, but never anything on the fungi.
Yeah.
Yeah, same here.
Yeah. I think the third one is what I saw in New Jersey. Yeah. Because I did not have my glasses, I just took picture I wasn't sure what it is because it was so tiny and so young. Um,
These are good
so
pictures.
yeah, I got a picture. So I know it's kind of a bird's nest, but I don't know which type. And this is really not mature enough to have that egg inside yet.
Do you I'm not entirely sure. Do you have to let them go to maturity or can you cut them open?
Huh.
Does anybody know?
You
I,
can cut them. You can cut them open, but the peridials aren't going to be mature. Um, so any spores you get from a mount are probably going to be immature and kind of unreliable. You want to let them open up, but it happens quick. You know, if you know where this is, check on it tomorrow.
All right, I'm looking at the thing when Jason was on here telling us all about the bird's nest. These are what we're looking at is the whole sporophores, and the top there that looks like the creme brulee part. There is the
yeah,
e-
that's a good way
e-
to.
epiphygram, a thin, delicate membrane or lid that covers the aperture.
Hmm.
And I'm not sure. I don't know my bird's nest all that well, but Nigellaraceae for sure. I'm not sure. The Could these be Nigella?
Somebody found some up at Stokes, just like this, still covered and everything. And I think that's what Sue was leaning towards. Uh, but she said if you take them home and hold on to them, they'll and I guess if you just kept them a little moist, they'll finish what they're doing and open up and the whole thing. So if you find some like this, I suppose you could collect them
Hmm.
and bring them home.
Yeah, pop them in a little moist chamber overnight.
Yeah. Yeah, I don't know if it's an overnight thing or it takes a couple days or what, but she seemed to be pretty adamant about take them home and they'll you'll get a better idea on them when they finish opening up.
Okay, next time I see them, I will bring back.
Yeah, and they're all over the place. I saw probably I look at I'm a nerd, so I look at parking lot uh, fungi in stores and stuff that I'm visiting, and
Mhm.
I saw this probably 3 or 4 times today. So
Yeah,
Yeah, what I,
ShopRite's loaded with these.
yeah,
yeah,
what I
yeah,
think,
Target around here.
yeah, what I suspect this might be different from what we saw on those grow on the mulch everywhere. This is actually growing on the real wood.
Yeah, I think these are the same ones that are currently popping out on my property on
Oh,
wood and on buried little pieces of wood, I think.
okay.
Um,
I'm
so, I think I've put a name on these before, but I forget what it is.
Mm.
By the way, I just sent an email. If we need more observations, I just sent some in an email because I was busy today. I just got around to it now. But anyway, let me look up the name that I think I had applied to things that I found in the past
it's
that look like these.
it's not Crucibulum laeve.
You know what? I think that's what it is. Actually, I think that's the name. Let me see what I have.
These are fun. Fun stuff. Thank you very much, Shihang.
I enjoyed it.
and hiking up above the tree line. That's you're real you really get after the hiking.
Yeah, it was really fun.
So, I missed your first observation, but was that also from Italy? The one before?
Yes,
What
yes,
was it?
yes. From my whole trip, I only saw two mushrooms.
Oh, okay, okay,
They said if you hike in late August, you will see lots of mushroom.
okay.
Um, and since I hike in June, it's beginning of season, I just see lots of wildflowers.
I see. But I bet that was so beautiful.
Yeah, it is.
Yeah, nice. Sounds great.
And Hunter, the first one she shared was Melanoleuca.
Oh, cool.
We
Nice.
we think.
Okay.
Does that anyone else have anything
Yeah,
to?
that I'll just say my pictures I just looked through. I just did a search on Crucibulum, and Crucibulum laeve is it looks like what Shihong had. So I think that's probably, but you might need to let them mature. I'm not sure if there's any difference between that and something else. Anyway, sorry.
Can you type in, please?
Excuse me.
Can you type in
Oh, the name.
yeah.
Yeah, I'll get that in a minute.
Okay, thank you.
You had the right genus, Shihong, Crucibellum, I think.
Okay. So Chris Ballroom.
Nice. Does anybody else have anything from their screen? We could go.
I have one thing. How many emails do we have? Just Dave?
Dave, but you know Dave, he's got 40 mushrooms in there.
Yeah. Okay. Well, I'd rather you share it as opposed to me sharing from my screen because that never goes well. So I'll send an email and I'll go after Dave.
Nice.
Chris, did you get the recent email of the lichen? I resent it.
I'm checking right now. Nah, Dave's showed right up. Still nothing from you though.
Strange, it says sent. I don't know what I'm doing. Taxonomy Tuesday, the usual place.
Yeah.
Weird. Sorry about that. I'll try
No.
see if I can correct it.
Maybe try putting it in the chat or something. I'm not entirely sure how to do that, but I know some people have the ability to do such a thing. If anybody wants to tell them how to
Is it?
drop it in the chat.
It's just a photo, not an observation, or is it an observation?
It's an observation on iNaturalist, and when I open it, it says sent, but obviously some glitch. I'll work on it.
Oh, you could put the whole address from the whole URL in the chat, maybe.
Okay, thank you. I'll see what I can do.
Nice. Thanks, Hunter. Hey, Dave, I believe these are yours.
Okay, yeah, and in the chat, I'm going to post in a link to Mushroom Expert for Crucibulum laeve. So, Shihang, you can just go to the chat and you'll see the link from DW. Okay.
All right, thank you.
Sure. Okay, I'm just going to go to full screen here. Oh, yeah, look at this. I found this yesterday or 2 days ago in forest litter, oak woods. It's some sort of Clitocybe, I think. Or if it's not Clitocybe, it's in one of these genera that's been recently split off. The interesting thing is, look at the bottom of the stalk. It's sheathed in this like dense gelatinous stuff. I don't know what to make of this.
Yeah, where'd you find this? Was it like in a swamp that?
No, it's in well, it was after a fair amount of rain.
Yeah,
The woods were kind of wet, but it was just well drained oak woods.
huh?
A lot of litter. It was growing in litter. I think it was attached to a stick, actually, a small piece of wood. It's pretty interesting, though. I sampled
Yeah,
it for DNA. Hopefully we'll find out something about it. But I'm not familiar with this sort of
yeah.
morphology.
The way that you described it was, I looked at it and I was like, is that like a gelatinous sheath? Is exactly what it looks like,
Yeah,
and that's how you described it.
it's what it seems to be.
Hmm.
Now maybe it's something growing on the mushroom. I
Or
don't know, but the mushroom
or
is in very good condition.
it is, and I'm wondering if maybe this thing had like uh, some kind of mycelium at the base, and that mycelium was maybe infected with some bacterium. Um, but yeah, I don't know. I don't really. That's cool.
Yeah, I don't know. I don't. There's no kind of like sclerotia type something going on with these, are there?
I not that I'm aware of in Clitocybe.
But this looks like it's coming out of it.
Yeah, it's really is surrounding the entire stalk. And I did something on my computer. I don't know what, and now I'm just see like your name, Chris, really big, and what we're talking about is in a small little picture on the side. What I mean, I don't know what to do here. Wait, I'm clicking stuff,
Can
not
you
the right stuff.
click on the window?
View, view. Oh, jeez.
Man, last week you were having a bunch of troubles too.
It's as soon as I try to do anything, I click something that I don't want to, and then I hide non-video participants. Standard. This well, is everybody seeing something other than Chris's name?
I'm seeing the spores, picture of your spores, but
Okay, yeah, I don't
the
see.
whole window is
Now I
small.
see IGS. I don't see anything. Yes,
Yeah,
I know. I understand that much. But I mean, how do I get an actual photo of to see what we're talking about here? Oh, there's the thing. There's what we're talking about in this tiny rectangle that's like of no use to any. Oh God. Oh, okay. I clicked on something and it straightened it out. I will forget in 5 minutes. Okay. All right. I'm sorry. I can now engage in discussion. So these are kind of interesting spores for Clitocybe also. They're kind of wide and not particularly lacrimoid, but they do have a big apiculus and they taper, at the apiculus. So I guess these don't look completely unlike Clitocybe spores. It's not a real good photo. I have a hard time getting photos of really small spores. The camera just doesn't really understand what it's supposed to focus on. But yeah, some sort of Clitocybe. I've saved it. It's dried out. It's in a bag. I sampled it for sequencing, so maybe I'll find something out about it. But yeah, I thought that was very interesting. That sheathing of the base of the stipe is just really weird.
Yeah, that is different.
Dave, just real quick, when you said you sent this for sequencing and it was vouchered and all that, is there a project going on, or you just sent it to
Well,
my coda?
we're in Pennsylvania. We have some money to work with, and we've involved Mycota Labs and Steve Russell, and so Steve's going to get paid some money, a fairly decent amount for sequencing a whole bunch of Pennsylvania collections. But he told me to send as much stuff as I want, as long as I do the labor,
Yeah.
which means fill fill the plates, fill out the spreadsheets, make sure I get good observations on them. But he knows I'll make good observations on
And when
wine.
actually, we'll talk about this maybe off off camera. But
Yeah, in a little while. I don't have all night tonight because I have some stuff to do later on, but I can stay on for a while. But yeah, so here's a great big Amidella, a really big thick one that you would think of as being whetstoneae. It's got the big tubular basal vulva. Somebody's already proposed whetstoneae, but I haven't even had a chance to look. I just saw in my email feed. There's probably Igor. Igor, that was probably you proposed whetstoneae
Oh
on
yeah,
Mushroom Observer.
that was
Yeah,
me.
yeah, it's probably what it is. But the thing that's interesting is the gills were readily stained reddish brown. Um, I don't know if I've seen that in Whetstoneae. Maybe I have, and I just forgot. But you see it didn't come through real good in this photo. Here you can see it a little better. Just where I touched the gills, this stain set in pretty quickly, and it was a little bit more pronounced than what this photo shows. I didn't want to try to mess with the editing too much, but the spores match whetstoneae also, but there's not a whole lot of variability amongst Amidella spores. They're mostly sort of not quite bacilliform, but I guess they're like ellipsoid, elongate ellipsoid. So the Q is like over 2 on some of these, or close to 2 at least. But yeah, it's probably whetstoneae. So of course I sampled this oak woods, everything oak woods. So on the 5th, I went to some oak woods not far from my house. I spent a couple of hours and found some things.
That's a nice estimate for sure.
Yeah, it was a great big one, really. I mean, it was not fully expanded, but it was still really big. I mean, you can see from the penny in the photo that it was pretty big. So we have this member, Grace. Her name is Grace. She's Chinese. Well, she's been living in America for a while. She's married to an American guy, and she maintains these little gardens surrounding her house. These like moss gardens, and these gardens with like exotic plants, and she's got these exotic trees planted on her property. And also, right on the border of their property is a great big oak. Oh, good. And this was in one of her gardens. So it's a Helvella. I think cupuliformis, maybe. Cupuliformis is typically not supposed to be this dark. The spore morphology kind of fit cupuliformis. I got weird spores out of these things.
It looks like, is it macropus or something?
Yeah, that's another one, but that one I think the size is a lot different. So I think there's a reason why I eliminated macropus, because cupuliformis and macropus look a lot alike, and there's a few others that look like that too. They look sort of like satellite dishes, and this is one of them. The spores kind of fit, but it's just pretty cool to find. I mean, during the period where it got kind of dry, I was able to go over to Grace's house and like find mushrooms, because she collects rainwater in a barrel, and she uses it to water her gardens, and she keeps getting all these mushrooms. Yeah, so you can see the spores are kind of strangely variable. I don't think there's anything growing on this. There is a hypomyces that grows on Helvella that I just found out about, because I submitted some Helvella last year to Steve Russell, and the sequence came back as hypomyces, because apparently that was the first stages of a hypomyces on the Helvella, and they didn't get a Helvella sequence out of it. But anyway, these spores you see in these pictures do not look like the spores of the hypomyces that favors growing on Helvella. So I don't know why these spores are so variable. I mean, it was from a spore drop, so presumably they're mature spores. Although I did read one account of Helvella cupuliformis. Maybe it was on a mushroom expert. I'm not sure. Where it was said that the spores mostly will have one large central gutule, but sometimes they'll have small gutules, and like two or three of them on either end of by either pole. So I guess that's kind of what I have here in these spore photos. It's just the sizes of the spores is pretty variable. So I think that's what it is, but probably not. Maybe just something very similar.
That's a cool find.
Yeah, it was. Yeah, I thought they looked kind of neat, so
That one's got
So
that like almost saddle type of
yeah,
thing starting to happen, or
yes, and some Helvella, I mean, INAT calls them elfin saddles.
is there a photo with a penny here? How big were
Yeah,
these?
there's a photo at a penny there someplace. The caps are about the
Yeah,
size of a penny, the biggest
yeah,
caps.
cool.
Yeah.
Yeah, the dark cap, the dark, I guess that's a cap. Is that the fertile surface?
The fertile surface is the concave part. Yeah, the upper part.
Yeah,
Because
yeah,
these are
part of the cap.
operculate
Right.
ascos. They fire off they don't have to be pointed downward for the spores to come out. They just fire them like cannons. The asci, that is.
But that part's dark
Yes, very dark inside the
for
cap. Yes,
this or what is it? Microporous,
macropus. Yeah, yeah,
macropus. Yeah,
it's
yeah.
dark. But
Hmm.
one of the either mushroom expert or Myco Quebec or the ASCO, the Bisset ASCO book, one of them does say that cupuliformis sometimes can be dark gray. But these are really dark. So I thought that was kind of interesting.
Yeah, I mean, I like it. Cool find.
Yeah, it was a good one. It was nice to find. So I preserved them and I sampled them. We'll see. So I got two sequences in a row, or two observations in a row, because I think they're the same species, growing about 10 inches apart from one another. There were a few of these little cups, and there's some micro to go along with it. And then the MO observation that's paired up with this one is probably the same species because the spores were very similar. But there's more micro. Oh, wait, maybe there's more micro in this one. Let's see. The spores are interesting. I smash mounted some of the material from one of these little cups, and the assay showed some of the really immature assay showed really fairly small spores that were kind of transparent and had two gutules in them and had no ornamentation and looked pretty smooth and thin walled. But these mature spores here, these have, you can't really make it out in the photos, but through my um, binocular scope, it's very clear that there are some very distinctive warts on the spores of that warts are about almost 1 micron high, and very you can make them out really well through the better microscope. It's just I can't get a decent photo through one of the eyepieces. So this is as good as it gets with the photos. So you can't really make out the um, the 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 warts on the spores, but they're there. But you can certainly tell though, just from these photos, that these surfaces are ornamented. But the difference between the immature and the mature spores is pretty interesting, and I think you have to go to the next link, which is just paired up with this one to see some additional micro. Yeah, from these guys. These are the same species, I'm pretty sure, even though the color looks a little different. The size is about the same, and they were about 8 inches apart. This group from those other ones. See, they're pretty small. Um, and I'm calling them Pezizales, but these are probably Pezizales. I should have mounted some of the smash mount in melters to see if the asci were amyloid on the tips. But you see here now, these spores right here, these are mature. I think these are from a drop. I think I even labeled the photo drop, but INET doesn't show your photo labels. But you can see there's like 2 gutules in there in the 2 spores up toward the top, and those gutules are kind of spreading out and becoming not real distinctive. And then in the other photo, you can't really see gutules. And in this photo, you can sort of see gutules. They're not real distinct, but you can see these spores are starting to develop the ornamentation on the surface. It's it's a little ragged looking. I think if we keep going here, we're going to eventually run into these photos that show the spores in a few asci, and that's where it gets kind of interesting. So I just took a whole lot of photos of the spores because I was kind of intrigued by them. That's like a very zoomed in close up. You can almost see the warts. So I think if you keep going, I posted a lot of photos of the spores. I think eventually.
Oh, there you go.
Oh, there we go. So look at the ascus on the left. The spores are almost mature, and they have the ornamentation. The ascus on the right, the spores are not as mature, and you can see through the wall, into the interior, and you can see these gutules pretty well. And the neck, and also that looks like there's a paraphis stuck in between, and maybe also a few other paraphyses. I guess that's how you say it, here and there, and they're kind of what's the word they use? Oh, I forget, but they're not clave, right? Because they're not really tapered, or they might be like very gently clavate. But anyway, here we go. There's another photo of spores in an ascus, and these spores are almost mature. And I think that there were only 4 spores in this ascus, so they're mature and they're popping out. Some of them have already popped out. Here we go. Here's a couple of photos of asci. And if you can zoom in on these, I'm not sure how much you'll be able to zoom. But look here, how small the spores look, how easy it is to see the gutules. The walls are apparently kind of thin. There's no ornamentation. And so the thing that's interesting to me about this observation is that you read sometimes about spores of some ascomycetes that are different when you see them in the ascus and when you see them outside the ascus, the mature ones. And it's the ornamentation a lot of times that's a lot different. And I think there's some Scutellaria species where this is a factor. And so that makes Scutellaria particularly difficult to study because, to see the mature spores, you almost have to get them out of a drop. And here, these are probably the most immature spores of all of the photos are in this one ascus here. And you can see they're pretty small. You can see the gutules very, very readily. The walls are apparently very thin at this stage. There's no ornamentation. So just kind of interesting, I thought, to see these spores in different stages.
Yeah, Dave, very cool. I think Scutellaria is a good example there.
Yeah.
I think Geoglossacea, the geoglossum type things, where you have to figure out the different segmentations, right? Because those
count the septa. Yeah,
septa, right?
Yeah, and sometimes you can nail a species name by there's one I'd have to look it up. I don't know my names offhand, but there's one where it's like always 7 septa. There's another one that's like always 13, I think.
Yeah, and on that note, in the asci, that is underdeveloped, so you can't make that distinction.
I always look at spores from those earth tongue things by lying one down on a microscope slide and waiting for some spores to drop.
And also on that last thing you showed, man, those asci were long and skinny. And I thought it was cool that the spores were just tucked right up on top of those really long asci.
Yeah. 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 Well, I think probably, there's some pressure inside the ascus that's pushing those spores up toward the that lid,
Operculum.
Operculum. Yes. Okay. Thank
Yeah.
you. Right. Yeah. So this is apparently some sort of species complex, Lactarius argillaceifolius. God, I think I actually said that the right way. So at least the Bissett book on milk mushrooms kind of leads you to believe that there's varieties. So the interesting thing here is, to me, I collected spore print for this one and for another one. I made two collections of these, and this one I put in for sequencing because it was a really nice. The fruit bodies are really fresh and very nice. And but if you read Myco-Quebec, in their comments they say, "Oh, the interesting thing about this species is the spore print is brown." Then you look in the description at Myco-Quebec, and it says the spore print is ochre. And then you read Mushroom Observer, and it says the spore print is yellow to ochre. And then you look in the Bissett book, and it says that the spore print is pale pinkish for this argillaceifolius. So this is pretty close to what this spore print looked like. It was like a dull ochre, pale brown. I did play around with the edit to get it to try to look like what it actually looked like firsthand. And this is pretty darn close to what it looked like to me firsthand. That's on a slide. It's a spore print on a slide.
I like how you threw all the options in there: pale ochre, brown.
Yep, there's all these different. So perhaps there's quite a number of species hiding under this name.
Yeah,
I don't know, but I sampled this one. It's hopefully the sequence will be successful and we'll find something out about it. I don't look like for cystidia usually on Russulaceae because a lot of times the cystidia are small. They're like hidden in the gills. You have to do like one of these really, really, really good Roman aqueduct things to see anything. So I usually just skip that. But what I do is, now I didn't do it with this mushroom, but so there's no description here of what the spores looked like. But what I do is I scope the spores in melters to try to assess the ornamentation, the warts, how big they are, and if there's any sort of patterning, and if there's any connections at all. I wasn't able to scope this one because apparently, I think some dirt got on the slide or something, and it wasn't lying down flat where I position it in my microscope, and I wasn't able to get the 400 times lens position to be able to see what was going on. So I just gave up because I had I don't know 15 things I wanted to scope. So I'm not going to mess around with one specimen for more than 5 or 10 minutes. But anyway, getting off track here. Yeah, the spore print color is actually sort of the interesting thing here. Also, the stain on the gills is from the latex. The latex stains the gills. It takes a while. You cut this. You make this slice on the gills, and you see the white latex, and then it takes a while. It's like a half an hour, I think, before I notice this brown staining. And a lot of the Lactarius, a lot of the milk mushrooms are like that. The staining reactions are slow. So, well, oak woods again. Pretty much everything oak woods the last few days. And it's slow. We had a whole lot of rain, but it's slow now because I think it probably has to dry out a little bit. we had some places near the New York, New Jersey, Pennsylvania border where there was over 6 inches of rain in one day. I think Stokes probably had a lot of rain. I think they probably had like 3 inches. So it's
Nice.
acrid. This tastes acrid. So it's not edible.
Thank you very much, Dave.
Sure. Thank you.
All right, let's see the mailbox again here. Oh, we got Brandon's, but I don't know what's going on, Doug.
I sent it as a photo and I can open it. It says sent Taxonomy Tuesday at ny whatever it is .org. Usual.
Yeah, I wonder if I could
I'm just on a little tiny iPhone. Maybe something's screwed up, but it shows it sent. I can open the sent.
Yeah, I just sent my email to Taxonomy Tuesday, 20 minutes, a half an hour ago, while the session was already started, and it just went right through.
hold on a minute. Let me do this real
While we're waiting, I was
quick.
late. Doug? What were those spores from that looked like bear aspirins or something? That you cut in half.
I'm not sure that was me, Dave.
Oh, that wasn't you. I'm sorry. Who was that?
Not sure.
I think it was Paul.
Paul.
Yeah,
Okay. Oh, yes,
Paul.
Paul. Yes.
Paul,
Yes.
did you share spores? I forget.
Excuse me.
Oh, yeah.
I'm sorry.
Those ones with the crusade or cruciate.
It looked
The
like an aspirin that you can cut in half because there was like a little line down the middle.
right. That was the basidia for the tremella.
Right.
Oh, that was from the Tremella.
That's what it was.
Oh,
yeah, it was really cool because it's the basidia, right, Paul?
That's correct. The basidia.
Oh, that was a photo of basidia.
That's right. Yeah. You'll see something similar if you pull it up on Mushroom Expert. Although I think my picture's a little better, frankly, but you'll see that's similar sort of.
Brag about it. Go ahead.
But
anyway,
anyway,
I'm gonna look into that. Actually,
it could be the spore is divided into 4 cells, maybe.
4 cells. Okay.
yeah, four septa.
Yeah.
4 septa.
So what do we got here, Brandon?
I mean, I'm calling it Ophiocordycipitaceae. I'm leaning Ophiocordyceps. I think starts with an S, but it's a whole clade. So trying to put it to species without sequencing is probably futile.
How do you know it's not some sort of Polypocladium?
So that's why I called it Ophiocordycipitaceae.
Okay.
Because Polypocladium falls into that family.
Okay.
Because I'm not sure if it's Polypocladium or Ophiocordyceps.
It looks like is that a bug?
Yeah.
Is that a pupae or something?
Yeah.
It's a cicada.
Cicada, right.
Oh, okay.
Which
I don't
is
think
interesting.
It's Polypocladium then. I think those guys, do they all grow on like deer truffles and stuff like that? Or
no, there are species, and I found species of Polypocladium on cicada nymphs specifically.
Uh-huh.
But morphologically, that that fruiting body looked much different than this.
Uh-huh.
This looks more like Talipocladium ophioglossoides.
Yeah,
Um,
a little bit, but that one wouldn't be growing on an
correct,
insect.
correct. Yeah, yeah, that would be growing on a Lamphomyces, a false truffle.
Yeah, I have actually a really nice observation of a what I think is Talipocladium ophioglossoides from a couple of weeks ago, where the head is like trifurcated. It's really weird. I have
Yeah,
to.
yeah, yeah. Yeah. So the funny thing that you say that because some of the things that I'm considering here, um, the head should have a little bit more ornamentation. One of which is, oh, what is it? Talipocladium. Um, is it paradoxum?
Oh, I don't know.
I
I
think it's
only know a few of these. I know Talipocladium longusegmentus, or Talipocladium capitatum, I think it is, and Talipocladium ophioglossoides. I don't think I've ever found anything else in that genus.
mm, well, I, I I like Talipocladium particular because there's more out there that meets the eye, and those are the three that are most commonly found.
Yeah,
Um, but once you get into the micro, they're actually not hard to deal with. Um, you can get some really good information, and all of a sudden, oh, here we are with Valiformi, which Rene Leboeuf named, or no, they didn't name it, but it's on the Quebec site, you know, and it's rare and it's found in New Jersey. I don't know what this thing is though.
So, so like, are we looking at all that was sticking up out of wherever you dug this thing out of?
Yeah, it was probably buried about to that notch, just under the obvious differentiation between the hymeneum and the infertile stipe.
And you did a hell of a job teasing that thing out.
Yeah, it wasn't easy. Look at all the
Just the
just
extra root there.
yeah, it's just extra root material. That was the toughest thing to excavate
Well, I think
through this thing.
those roots are probably from like whatever tree or bush is nearby, or do you think that belongs to it?
What, the fungus or the bug? No, 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 no. That's just yeah, stuff growing underground, plant material.
Yeah, you nailed digging this thing up. You did.
how long did it take you to excavate this entire thing?
It was found in a muddy area, so
Oh,
I was able to get a clump of soil up, right? But
yeah,
then getting that clump of soil and not ripping the thing apart, it was easier than I thought because these are sturdy stromata, right? Talipocladium or
uh-huh.
Ophiocordyceps. Um, they're sturdy stromata, and there's a good attachment to the host. But all the other stuff, yeah, it did take a little while getting that off.
Congratulations is all I got.
Hmm, hold on,
Yeah, that's a good one.
I'm looking for that name. It'll come to me. But yeah, thanks. That's all I got.
Thank you. Let's see. mailbox here. Still nothing in there.
Doug, can you just paste the URL into the chat?
Is anyone else in line? Because I might have a few things to show.
It sounds like Bianca. Go for it.
Yay. Okay. Let's see if I can figure out how to share this again. This way. No, of course not. Start share. From photos. Okay. This is from today at.
We don't see anything.
No, I'm getting them. I'm just mentioning it before I get it. Yeah, so we'll start with a zoomed out, just seeing kind of a lot in the same area. Let's try. Can I do all of them? Oh, hell yes. All right, we're just going to do this. Oh, did it do it? Oh, let's try that. Okay, I think I'll be able to go through them. We'll see. So this is kind of away from them. You can see a couple of mushrooms in the area. One that was toppled before I had even gotten there. The one next to it, the little one on the right side kind of has, let me see if I can zoom in, you can see it, this guy on the right here. There was also another one slightly more to the left. So they're kind of sort of spaced. I think this is the cap of another one in the same species. All right, so this is zoomed into one of the, that's the other one that's not pictured. I think it's also in the same grouping.
Amanita amerirubescens group.
Yeah.
All of them?
All of them.
Yeah, they're all rubet. Look at the inside. You see the staining going on.
Yeah.
The flesh.
Cool.
I could even see the staining from the small picture, the first one that you showed from far away. I'm like,
That's amazing.
There's got to be amerirubescens, yeah.
With this one?
It looked like it even from that distance.
Cool. Very cool. My first time finding this many in one group.
Yeah, you can eat them after boiling. Dave is going to tell you all about this.
Oh, amazing!
Oh, there's more than one species in the complex.
Oh yeah,
a long time ago, I ate some rubescent amanitas, and I thought they were really good. And then I ate some more, and I thought they didn't taste very good. I probably sampled two different taxa.
Must be different species or something. Yeah.
Yeah.
Okay, well that's cool to know.
Basically, you need DNA to figure out what species you have, and nobody does that because these are so common. And who cares? We just call them amerirubescens group or complex. And
I'm going to put a few in for potential sequencing.
Yeah. But
I've put a few in the past also, and I have got a few different species names.
Yeah, so do I. But you can sequence all you want, and you'll never be able to put sequencing data and morphology together to come up with decent species concept. There's a lot of overlap. They're too variable. They're too overlapping. It's just a mess.
How about the one with the yellow PV though?
There's two of them at least. I think
at least two of them. Okay.
Yeah, there's two of them. Yeah.
All right. So this is moving on. I was wondering what this red staining was on the lichen. There was a bunch of the green lichen on this area. It looked like it was either dying or maybe I thought might have been something like a hypomyces for lichen. I don't know if that's a thing.
From this distance, I would just say Sterium complicatum. Can we see a?
No, as I understand it, that is pretty much what they look like when they're dying. The red, a lot of these, shield lichens, Parmelia type deals, um, they they go that way when they're
Okay.
not doing so hot. And
Okay.
that's only my understanding, so I
Well,
could be wrong.
what about this black dottedness in the middle here?
That's a fungus,
Oh, okay. I don't
I
have a
think.
better shot
Oh
of
yeah,
that.
that's like a hypoxylon type thing, I guess. Jack Rogue Sorella.
Mm,
Well, the
So,
reason why I asked if it was part of this
yeah,
dying
maybe.
fungus is because of the ring. Do you see that ring to the upper right?
Hmm.
This, I thought maybe they might be related. Is that not? Do you think the fungus might be killing the lichen?
No, I think maybe it looks like some of the lichen was damaged in that circular pattern.
Hmm. Okay.
Maybe.
Maybe.
Just a theory.
Or it just never grew there.
Or just
Okay.
never moved it. Yeah.
Gotcha. All right.
So
Moving
it's
on.
a dying lichen, so we're going to propose a name, lichen kaput.
Perfect.
I do
Um,
think that's a natural thing with lichens. Yeah, who was just about to? Yeah, go ahead.
okay. So this, I think, is crown-tipped coral.
Yeah.
yeah, it
Nice.
looks like it has the little circle of little fingers on the end of each branch.
Artemisias.
Oh yeah, yeah,
Yeah.
And I've
that
heard.
one
Formerly,
photo you could see.
formerly Clavicorona. Yeah.
Oh, okay. That's the old genus.
That's the old genus, yeah.
Okay.
The
Yeah.
invalid genus, yeah. Yeah.
And I heard this was edible. Am I right?
Yep.
Yes.
Cool.
Yeah, people eat it. I don't think it's that good, really. Some people love it.
All right. So I took that piece that we're looking at, that the only one that looked like it was not browning. Um, so I wanted to just, for curiosity, try it. Any suggestions? I'm thinking just butter on a pan.
That should do it.
All right. We'll
Cook
do that.
well.
Cook well. Got it. Thank you. So this, I have a couple of photos of this. I used my my clip-on micro lens, but you can see outside. Very rotting wood. I can't tell you what it is. And then here is a zoom in with the micro lens. Thought that was pretty cool.
Nice.
Cool.
So what's that? Serratium?
So
Mixa?
Fruticulosa or something like that. Yeah.
Yeah, it's the noodle one. The one that looks like noodles.
Oh, there's
Oh,
another
nice
one. Yeah,
photo.
right.
Yeah.
Oh, that's nice.
Yeah, I'm very proud of my $20 purchase for 3 lenses from Amazon. And this is one of the lenses of being able, I just take it around in my little purse with me just in case I'm doing this stuff. All right, so
Yeah, that's pretty good because these are hard to get a good photo of. Because usually when you have like a lens that magnifies a whole lot, it's only going to be accurate like within one plane.
mhm.
But it looks like you got a little bit of dimensionality there,
Depth of field.
depth of field. Yes, that's what I
Yeah,
was
that's
searching.
a that's a photo contest entry right there.
This
Oh,
one?
yeah.
Oh, okay, cool. Good to know.
Definitely.
Thanks. All right, let's go to the next one. So this guy kind of looked like an oyster, but I wasn't sure. Let me just show you the underside. I don't know if I have a better shot than that.
Epidotus, it looks like to me.
Crepidotus saponatus or whatever.
Well, I think we want to see the entire thing because it looks like this one's missing part where it was attached to the
Oh,
substrate.
well, I mean, that's kind of
Yeah, because it could be Hohembiohelia angiostatum. It could be. It does look a
I
little more
kind of
like
just ripped it off.
Crepidotus.
You can
You
see it's
could
just
tell from spore
exactly
print.
that piece. Spore print?
Yeah, Crepidotus all have brown spores.
Oh,
Hohembiohelia has white spores.
I have
Oh,
a feeling it's white spores because I can see there's powder. Oh, where is it? There's like a white powder on this top one, I think, unless that's the mycelium getting
yeah,
onto it.
hard to say.
yeah, these
Hard
could
to
be
say.
just young. They haven't browned yet.
Oh, okay.
Yeah,
Okay.
it could be immature Crepidotus, right? Yeah, I'm not going to commit to
Okay.
either genus here. But in the
Shoulders are
background
very
here,
common.
it looks like some of those caps kind of taper to not quite a stem, but sort of like a stub
Yeah,
that
I
goes
could
into
say
the
that.
substrate. See that to me looks like Hohenbiohelia androstetum, but it could be a cryptotus.
Hmm. All right, so I'd have to go back when it's slightly more mature and see whether it made
You know,
brown spores.
if you had brought these home in this condition and got spore print, I'm sure you would get a print. These look mature enough to get a print.
Okay. They have the. All right, we'll move on. So I found this. It kind of looked like a scleroderma, but I've never seen one orange like this. So, and I did, so that's the size. And then I cut it in half. So this is the opposite side before I cut it. And I zoomed in a bit on that. So it was hard, but I cut it in half and it had this white,
Oh, neat.
which is not what I was expecting at all.
Oh yeah, we found something similar to this at an NGMA foray. It was some excitement. I forget the name, but I think we've seen this before.
Okay.
Some kind of a
Gotcha.
truffle-like thing.
Yes, yeah, it kind of seemed like a truffle. It didn't have a particular smell that I remember. It was
Yeah,
kind of tiny.
yeah. I remember Nina produced the name. It was orange on the outside, very bright orange like this. I have to ask Nina because other than that, I just cannot remember what it was.
Gotcha. Okay, so I have a couple more, but I want to give room for if there's other people that want to share before I continue.
Well, go ahead. Doug's thing came through, so after you go, we'll bring his right up.
Well, why don't you go with him? I have a bunch, so feel free to go with his, and then I can continue after.
Circle back.
All right, let's.
There it is. Okay, Chris, can you hear me?
Oh yeah.
Everybody hear me? Yeah, a couple weeks ago. Sorry about all that confusion, Hunter. Thank you. I just went at iNet and pasted the URL, and that solved the problem. I think my old iPhone 13 is dying. Need to replace it. So, 2 weeks ago, Chris, you asked me if I had anything. I didn't really have anything interesting, but I said I had some family up in Alaska. I'd asked them to keep a lookout, and they sent me a couple of photos. This is a lichen we won't see down here because it's restricted to Arctic tundra. It's kidney lichen, and there's apparently about 9 or 10 species, according to Brodo's book. There is one that occurs in the East Coast called fringed. It's Nephroma helvetica, but it'd be pretty far north. It was taken near the town of Bettles, a population of about 30, which is way up north, Arctic Circle, right below it, below the Brooks Range. And it was founded as a gold rush town in the 1890s. And now it's not much. There's no way in except by plane or water. And it's a gateway to the Arctic National Park. So this lichen is unusual in that it's tripartite. It's got two algae. It's got a green algae called Coccomixa, and it's got nostoc, the cyanobacteria, which is a nitrogen-fixing bacteria, both. There's another species called the alpine kidney lichen that is found up there that's also green. The other 8 only have nostoc. This is not the alpine kidney. The lobes are smaller and it's different. There's no ceridia or isidia on the thallus. You don't see the apothecia here, but if you look closely in the middle, you see these dark little dots. You see them on the upper part of the green thallus, not the margin.
Yeah.
Yeah, those are cephalopodia. They're special structures for the nostoc, the nitrogen fixing bacteria that take nitrogen out of the air. And in these nutrient poor parts of the tundra, apparently this is a very common lichen, and it contributes significantly to nitrifying the poor soil.
So I
Unfortunately,
know in
Wow.
like bean species or whatever that associate with a nitrogen fixing bacteria, they make these nodules.
yes, yeah, those
So are these spots like their nodules?
they're invaginations. I couldn't find a microscopic picture of it. The bean stuff is rhizobium. It's it's a totally different. It's a bacterium. It's not a cyanobacterium, which are very primitive nitrogen fixing bacteria. Um, it has these wavy margins. It can be pretty large. You know, these thalli are several centimeters across. You can see the reindeer lichen in the background, and you can see some moss there. And I looked in Brodo's book, Lichens of North America, and he says that this is eaten by the indigenous people. Even now, they mix it with crushed fish eggs. They boil it and mix it, and the caribou eat it. It's how it tastes. I have no idea, but I thought it was interesting, both from well, we had someone talking about the Dolomites. It's far away. It's way up north, and it shows how lichens are very important parts of ecosystems. The tundra, unfortunately, that you all read about is melting. You know, some of these villages out toward the coast now have to be moved at government expense. And that's a real issue with the warming of the tundra. And but it's kind of an interesting, it's quite beautiful with the lobes and the wavy white edges. I wonder if I have anything else to say about it. When I looked in his book, the apothecia are bright red, but I don't see them. The spore-bearing structures are, you know, cup-shaped, typical. But this particular photo that was taken does not show them. Has anyone ever seen a nephroma lichen anywhere? Maybe they could comment.
I don't think so.
No, my wife observes lichens. She's never seen this genus here in Pennsylvania or New York or a little further north even.
Yeah, I mean, most of them are pretty far north in the Pacific Northwest. Um, I'm looking at Broda's book here. He does show fringed kidney lichen, Nephroma helveticum, actually coming down into our area in Appalachia. I'm not sure. He says on mossy rocks and bark, especially in humid forests, stronger tolerance for dryness. So apparently it does make its way down here. All the other species seem to be way up northern Washington State through the Cascade Range into BC into southeastern Alaska, and they are brown. They only have the nostoc. There is something called naked kidney bean lichen that looks like it comes down into the southern Rockies. So that would be the only other one. This one does extend all the way over into, according to Brodo's map, uh, into Atlantic Canada. So it's possible that someone could see this if they get pretty far north on the East Coast. And I don't think there's anything else I have to say. It's kind of interesting with the beautiful color with the green algae there. And I don't think it's that common for, I know one of us, I forget who it is, maybe Jason is a lichen expert. Do many other genera have both more than one type of algae as their photobiont? Do you know?
Was he even here?
I'm not sure it was him. There's somebody knows a lot of lichen. Who is it who does the lichen?
Oh, it would be Jason.
Yeah, it is Jason.
Jason, or it could have been Dorothy also. She knows a lot about lichen.
Yeah, yeah.
But no, I don't think there's more. It's very common for there to be more than one whatever, lichen or cyanobacteria associate. I think it's very few that have both, even.
That was my impression from reading Brodo's book. I couldn't get a whole lot on the just searching. The U.S. Forest Service has this very nice write-up about this particular species, so maybe there's more information referred to in there. But I thought it was kind of fun just to show something from way up in the frozen. It's not frozen anymore. I shouldn't say that. It's summertime, way up north.
Yeah, that thing's beautiful, man.
It's very interesting.
And thank you for bearing with me, Chris. Sorry about not being able to get the.
No. Technology's for, headaches only.
Yes, I'm discovering that. All right, thanks.
Thank you.
Yeah.
Did you have some more there, Bianca, you wanted to?
Yes, a few more, but I didn't want to, if other people are in line and want to share, I had plenty to share this. So if you want.
Go right ahead. I think the floor is yours.
All right. Start share photos. Let's do. All right, so I went to the crown tip coral, so I'll do 3. It started getting darker, so the photos aren't going to be as vibrant, well, as clear. I can choose so many. I'm so excited about this. All right, yeah, okay. There's a lot to get through, so I'll be quick. If you know what it is, just shout it out.
Tricaptum.
Yeah, it looks like tricaptum.
Correct. All right, so then this guy.
Oh, I may have found this in my yard today. Something that looks just like this. I didn't know what it was. I took some photos of it. I haven't posted them yet. And I sampled. And I'm seeing if I can get some spores. I think this is probably a brown spored thing.
A brown, say that again?
Brown spore print.
Oh, brown spore print. Oh, okay.
Yeah, I'm guessing for this.
I haven't.
I don't know what it is, though. I think it looks like the thing I found today. I mean, if it's white spores, it's probably a mycena, but I don't think this is mycena.
Maybe Boletus?
I suppose that's a possibility, but it doesn't really say Boletus to me.
Can you zoom in on the margin of the cap?
Is that Boletus, I guess?
Yeah. It looks like marginate velar material.
Yeah, I thought maybe it was a
that might just be
Tremella.
gills. That's just gills, I think, that you see.
No, I don't think so. Here.
No.
Oh,
Here.
it is. It is. Okay, it is material. Yeah, so this probably had some kind of veil.
Sapphirella-like. Yeah.
Yeah, some sapphirella, yes. Yeah, you think.
It has a hollow, fragile stipe, as far as I can see.
There's the stipe.
It just snaps.
Yeah, I would think it would, yes. I think so.
Yeah, so.
Yeah, sapphirella are really fragile.
All right, I'm going to scoot next. Then there's this guy. So bear with me. It was a very dense mushroom. This is you could, that's not as great of a shot as I wanted. Maybe you can see a little bit there. This was a very young, very dense mushroom. And I did cut it. Yeah, you can sort of see the gill structure there, but it's very strange, almost worm-like, but I think maybe because it's young. So that's me cutting it in half. It has a little bit of gill to it.
Did
Any
you
thoughts?
observe any liquid coming from
I don't have any liquid. Sorry.
Looks like
any part that was
Crucibulum, either
cut?
Russula or Lactarius.
Yeah, it's Pezizales.
I'd be a little surprised if it was a Russula with how dense it was. It would withstand a bit of pressure.
There's some Russulas that are very dense.
Oh,
Especially
okay.
when young,
Yes,
they don't get fragile till they're older.
So I think flocculosis, Russula flocculosis is one that's really dense. I found a Russula today that's similar to this, and it has the really tight gills, and it's very, very dense. And I haven't posted it yet. But I'm gonna get flocculosis off of Michael Quebec and put it in.
Isn't there staining on this thing? I thought I saw some brown staining in the other photographs.
Let's see.
Yeah, like this. Maybe this is just compacta.
Well,
it didn't smell funny. I
was going to bring up compacta when you were talking about,
Yeah,
or Russula all fragile. No, they're not. There's many tough ones.
It's injured on the stipe and it stains kind of red in there. And compacta does that. The gills stain brown too.
Okay. I'll keep that in mind.
So that's a possibility.
Here's the next one.
There are also some red stainers, and there looked like there's some red staining going on in that.
Well,
and they're also really tough. So, and they tend to have,
yeah, this doesn't look like one of those red staining ones. I found flocculosis. Oh, here it is. This Russula flocculosa is really variable, but it does have very dense flesh.
All right, here's the next one. I think this might be the reddening lepiota.
We have to see the cap. Could be enough.
Oh, I think I moved too fast. Sorry. Darn. I don't have it. My bad.
Could be that just amerirubescens Amanita. So.
Oh, okay.
We have to see the cap.
It does have the veil. All right. So what would be the cap difference between them?
We just have to see the cap to whether it's a lepiota or an Amanita. There's going to be a difference in the disposition of the scales, the colors, etc.
Okay. I gotcha. Yeah, I'm sorry. I thought I had that picture, but it doesn't seem, unless I didn't click on it when I was choosing the photos. I'll check in a second. Let me get through the rest. So there's this guy.
Yeah, it's probably Xanthoconium athene.
Oh, here's the cap. I did get it. That's the bottom. Hold on. Wait a second. It must be out of order then. No, that's this one. Oh no. Killing me. Got excited. Sorry. All right. Xanthoconium.
Xanthoconium athene is being already parasitized by the mold, Hypomyces. Yeah, I've seen some other pictures, and this is like a typical appearance of that species when it gets attacked by that Hypomyces. It just gets malformed, club-shaped, poorly developed pores, tubes. So yeah, this species gets frequently attacked by Hypomyces. So that's what I think it is. It's just Xanthoconium athene is being parasitized by Hypomyces. What is it, Dave? Chrysospermis or something like that?
Chrysospermis is
Yeah.
usually what I find on these Epiconium.
That would be my take on this one.
Yeah, it looks like it's in the early stages, so you only see the white.
Mhm.
And then a little bit later, it'll turn yellow.
You see how malformed the tubes are already?
Yeah,
Just-
that's what this is. I
It just
agree.
the infection goes for the juicy parts in the first effect.
Okay. All right. So this was a very vibrant rusula that I saw in the distance.
Yeah.
But I was surprised how long the stipe was. I normally find them much shorter.
Yeah. Did you taste it?
I did. It had no taste, and that's how much I was able to peel down.
Dave, does it look like rugulosa to you?
Well, it does, but there's so many red russulae. Some of them are acrid, some of them are bitter, some of them are mild.
No, but there's a Rusula rugulosa thing that is.
Yes, that's one of them.
There's more than one rugulose rusula.
No, there's just more than one red rusula.
No, but I was specifically referring to Rusula rugulosa.
Well, it could be that. That's one of the possibilities.
Right, but
there's so many rusulas that look like this.
No, but very few rusulas have really rugulose caps. I thought that one was distinct.
Oh, okay. Yeah, well, that one has. I think it has also a striate tuberculate margin. I'm pretty sure.
Can you zoom in?
It is mild. It does taste mild.
Yeah. You see how caps are rugulose? The skin? And it's tuberculate striate on the margin.
Yeah, it's got a grooved margin. It looks like it might have a little bumps too.
Yeah, lots of red rusulas.
Lots of them. Yeah.
Okay.
Your only hope is sequencing.
Okay. I'll leave that to the
maybe
professional.
We're sort of lucky we get a temporary code like Indiana 55 or something like that. Just making up a number.
All right. This one I was actually pretty excited about because it's not what I expected. So this is what I unburied. And then it had a partial veil, which I wasn't expecting.
Yeah. Yeah. Yeah, it's an Amanita from section Valde. I think this is possibly submaculata.
Yeah, could be submacula, could be brunescens.
Could be brunescens. But
yeah.
Submaculata, it stains red too in the stem when you injure it. But it has a big white veil, which this one does. Brunescens has a cleft-footed vulva. This one.
Yeah, that's what to look for, and it looks like it's got a cleft in it. See on the right there.
Could be brunescens. Yeah.
I think this is brunescens.
Yeah. Now that you sectioned it, we can see the bulb better. Possibly brunescens.
Yeah, submaculata doesn't have the really abrupt bulb like this.
No, it's more oblong and it has some fluff on it.
Yeah.
Here was one that was growing behind it. So there's.
This is.
There you go. There's your
that's
classic
unambiguous.
Brunescens. Yeah.
And it was a little bit of slime on the cap too. Is
a little bit viscid. Yeah, when they're wet especially. But look at the big cleft in the bulb, the big split. There's probably one or two other splits as well. They call this starfoot Amanita, I think maybe on INET. I'm not sure. It's one of the names they call it. Yeah, because the bulb splits apart and looks like a star a little bit.
Yeah, it looks like clefts. I just call it Bruno the cleft foot. I call it.
Okay. We don't talk about Bruno. All right. Sorry. All right. So then there's this. It was like a crust fungus, but I was able to pull off. See that the lighter one near the bottom of the screen here. So this one, I was able to pull that one off. And that's the underside of the bottom of that one that I pulled off.
Sterium complicatum.
Yeah, that's what I think this is.
Me
Yeah.
too. Yeah, unless it bleeds, right? And then it's the other one. If it has like a
Oh, that one that bleeds. Yeah,
which
what's that
starts with a
called?
G.
Ossipatum.
Oh,
Gossipatum. Yeah, thank you. Yeah.
What's the species name?
Sterium gossypatum.
Oh,
Is it?
Gossypatum. Yeah.
Yes, yeah. That's at least close to. Yeah.
I never find that one though. They never bleed for me. But yeah, so it's probably complicatum. It's really pretty right now here too, and I always think of that as something I see in the winter, but I'm seeing a lot of it now too.
Yeah, after a rain, it brightens up.
Well,
okay.
that other piece that you showed that's more light in color, that looked like something else to me.
Well, that's the underside. Literally,
Oh, okay. You just flipped it over. I got it.
yeah,
Okay.
it's the piece that's on the bottom there, that lighter one.
Yeah.
That one I was able to pull off and flip over. That's what that
Right.
is.
Yeah. So the fertile surface is completely smooth.
Yep.
And therefore it's a sterium.
Okay, very good. And yes, it's rather smooth. All right. Let's see. Moving on. Then this one. This is me. The coin is actually vertical, so it's kind of leaning on them. This is on a tree that's still up, even though it's definitely on its way out.
Oh, that's Trametes.
There we go. I wasn't expecting this color for Trametes. It was much thicker than what I was expecting Trametes to be. Is there a thicker Trametes that it's almost like a rubbery?
Yeah, there are big
Yeah,
Trametes, like Tinea. And there are smaller Trametes, thin.
Okay. All right. Well, I want to show off how good the camera was, that little lens. I was able to get this shot of a jumping spider
Wow.
going
that's
close
great.
to this. That I was so proud of this.
He's
And
so
it didn't
cute.
move. Yeah, he's super cute. All right. So then on my way home in somebody's front yard, I found this guy.
Oh.
Very dense.
Yeah, that's
Darkelii.
Bonderzevia.
Yeah.
Bonderzevia berkeley.
Yeah.
Oh, the Berkeley eye. I finally found it. Okay, cool. Got it.
Have you eaten this before?
I have not.
Yeah, be careful. Some people have a reaction to this thing, and
some people have pretty bad reaction to this.
Yeah, they
I've
spend
only
a few
eaten
days
this
on the
once.
porcelain.
Real
I'll
young.
skip it. I'll skip it. It was older. It smelled kind of funny. I just decided to leave it.
Yeah, this is the thing to avoid. Otherwise, we'll be talking to the porcelain maid for a long time.
No, I'm good. All right. Also, in this park, I don't know if you guys know, there is a passion fruit plant growing wild.
Oh,
whoa.
Oh.
Nice. I love those. I've never seen one in the wild though.
Is this a rarity in New Jersey or something?
I think
No,
there is
actually,
a native variety.
Yes. I did look it up, and it does fruit, but it doesn't produce seeds as far as I was able to tell. I did cut one open in the later season last year, and it just was, it wasn't, you would expect a passion fruit to have like a yellow sliminess in it. That's the actual fruit you would eat, and there was nothing. It was dry, kind of like a cucumber without the insides of it.
What park is this?
This is Shark River Park, and the trail that I normally take is the River's Edge is what it's called. And this is
I've been down that trail a million times. I've never seen that.
well, this is not on the trail itself. This is not on the trail itself. It's on when you come from the parking lot and you go into the big field. If you go straight the way that if like a maintenance vehicle would go up through like onto the field, and then you would turn right, it would be on your right side as you would go towards the river's edge.
That's awesome.
Yep, very nice. I love the look of them. Very exotic looking, and that was the underside of the Berkeley eye.
Yeah.
I don't know why it shows out of order. Sorry. But yeah, there's a ton of this growing in this one spot. You can see all the flowers even in the background. There's also I think it's over here, and there was a ton of it growing. I think it was a eggplant relative, but I'm not sure exactly which one it was. Okay, that's it for me.
Yeah, I thought you would be showing more boletes, but I guess they're not there yet.
Nope, not here yet.
You know what, Igor? I'll show mine from Shark River.
Ah. Yeah, I thought Bianchi went to Holmdel. Oh, okay. Yeah, I think I know what this is. Yeah.
This was immediately, and then this is about 5 minutes later.
Right. Yeah, I think this is one mile of what we used to know as Polydorosia. Now it's rubissima. Yeah, the staining is subdued. Bicolor doesn't stain usually at all, but this is showing some blotchy bluing. Yeah. This
So
is
is this the one that, because it's funny, because I found this, and then the same day I listened to that lovely presentation by Colin Dumnauer. Is this the one that he was talking about? Is the same as the Asiatica then?
It's the closest relative. Yeah. It's the closest relative from North America of Van Maua asiatica. Yeah. So this is not like unheard of when our fungi are more closely related to something from Asia as opposed to from North America. So this is just one of these cases. So yeah, I'm pretty sure this is Van Maua pallidorosea, aka rubissima now. Yeah, and it should smell like beef or chicken bouillon if you smell that one.
It had a something, but
It had a something. Okay. Well, you have to get more experience. Yeah, I'm pretty sure that's what it is.
Is this
cool.
edible?
It's edible, but people just say it's just, not much flavor in it. Well, maybe if you eat it raw, you're going to see little man, and then you're going to write a memoir about that. Yeah, it's perfectly edible. It just doesn't really amount to much, according to most people. I might have eaten this before when I thought they were bicolor back in the day, maybe like, 16 years ago, 17 years ago. Yeah.
I love the smell of them. I think it's so appealing, I think.
Yeah, the smell is all right. It's I wouldn't say it's like great, but it's not offensive.
To me, yeah, it's obviously a personal thing, but I just like it.
Yeah, they have a very dense flesh, and it's typically not very yellow to begin with. And as they grow older, it's almost like white, and the staining is typically very, very subdued and not continuous, but like in these areas, which this one demonstrated. And this one doesn't have the gestalt of bicolor, which we did find at Shark River when we were there 2 weeks ago. Or, yeah, it was 2 weeks ago, right? I think so.
Yeah.
Something like that. Or maybe 3. Yeah.
Cool.
Right. And if the bluing didn't turn into this kind of a bright reddish brown color, then you have polydorosia. If it did, it would be
You know what? Like the next day, they had completely gone back to the yellow.
yeah. Then this would not be pseudosensibilis, which we can rule out just by the looks of it because it doesn't have a brownish or tan cap. This one clearly has a shade of red. Rose red or whatever you call it. Yeah, pink red cap. Yep.
Thanks.
Pretty certain of it.
And I had some other mushroom weirdness here. Let's see.
Some weirdness.
Is that just playing small? Let me. Oh, there we go.
Oh,
Some auricularia, like 14 foot up in a tree.
yeah. Auricularia angiospermarum, most likely, because I think this is some kind of an oak or a hardwood at least. And allegedly,
And then,
these are good edibles.
Is that the kind that they put in the hot and sour soup?
Yep.
Nice.
And back in the day, I used to go to this Korean supermarket, and they were selling fresh auricularia from, I guess, China or Korea. I don't know where from. And I ate them. They were great. Cooked, obviously, not raw.
then the just that one, the spores coming out of them.
This is from Stokes, right?
Yep. Yeah.
Is that Ganoderma?
Yeah.
Correct. Tsuge. Yep.
What else did I have from there?
What's the one on the upper left?
Oh, I actually have a picture that goes with that one.
They're
Quite
disturbing,
squishy indeed.
Yeah. Almost like jelly-like, right?
Yeah, but it had like this like pore look to it. And I get that.
Yeah, it looks like a mess, but a beautiful mess.
Yeah, I'll have to find that picture and play that again like next week.
Any idea what this is, folks?
No.
Yeah, this is probably trichaptum.
Yeah, tricaptum.
Tricaptum, yeah, biforme, or whatever else is out there.
But boy, was there a lot of it. And this one I found the little pins on a
Yeah,
bunch of the caps too.
right. Oh, you forget the pin's name. A long one. Yep.
Phagophilicium.
Right. Thank you. Yeah, I read they're not parasites. They're just scavengers. They
And
don't
then
hurt.
this is back behind my job again this year. These are brand new. These are just a day or two old. This is the old one from last year with all the fused bodies.
So what is this again?
It's a Ganoderma curtii.
Ganoderma curticeae?
Yeah.
Oh, yeah, but the picture before that showed two light-colored mushrooms. They were
Yeah.
not.
Puffballs or
That's
something.
them.
Oh,
Oh, really?
Yep.
They're that
wow.
pale?
Yeah, I have a newer picture of them from the other day.
Huh. Yeah.
They're just brand new.
Wow. Looks like albatrellis or something.
Yeah, they
I thought
didn't
they were.
look
Oh
like
wait, this is the newest picture.
Ganoderma. They're pretty pale.
Oh, that's so cool.
Well, they just haven't developed the yellow and stuff yet. I mean, they're just now starting to.
Right. They are sort of they haven't developed the pigments probably because they're young and they haven't been exposed to the sun, right?
I mean, well, it's been since they popped up. It's been pretty cloudy every day, but these two are pretty close to each other. They're going to end up fusing together.
And then you have the Virginia creeper next to it.
Yeah.
Yeah, cool. Curticeae.
Can you
Yeah,
update again
like
next
every
week?
year.
Update again next week. I'd love to see the color change.
Yeah,
Yeah.
I'll keep after them. Now, does anybody else have anything to show from their screen? Otherwise, I will stop the recording. If I could find out how to do. Oh, there we are.